and diagnosis. Before we begin, I'd like to share a few housekeeping announcements. Please ensure your mobile phones are set to silent mode throughout the session. Toilets are located at the east wing of level 1 near the gallery. While refreshments are available throughout the day outside this room, kindly note that eating is not permitted inside the session rooms. This includes coffee as well. Okay. Do take time to visit the exhibition booths located at the pre-function area and gallery during the breaks. Don't forget about the lucky draw. Please check your goodie bags for the lucky draw card and collect all booth stamps to be eligible. Sunway University practices a strict no plastic policy, so we encourage everyone to avoid using single-use plastics. Water refill stations are available across the campus. Please bring and refill your own tumblers. There are several side events happening throughout the Congress, and you're welcome to join any that interest you. Lastly, we would like to humbly request all participants to kindly hold their questions until the panel Q&A session at the end of each plenary session. Okay. This morning's agenda includes 3 invited speaker presentations focused on current advances in cancer screening and biomarker research, followed by a panel Q&A, and then a series of rapid-fire presentations highlighting emerging studies and early-stage research. Now it's my pleasure to introduce our track chair for this morning's session, Dr. Jason Chin Yu On. Dr. Jason Chin is the operations lead of the Care Outreach Department at the National Cancer Society Malaysia, where he also runs the Quit Smoking Clinic. He plays an active role in cancer education, early detection advocacy, and patient support nationwide. Certified in basic life support and smoking cessation, Dr. Jason contributes to key national initiatives such as the Roadmap Towards Downstaging of Breast Cancer 2025 to 2035 and the upcoming National Strategic Plan for Cancer Control Program 2026 to 2030. His interests span public health, community engagement, and oncology research, particularly the effects of smoking on cancer formation, treatment, and recovery. With that, I'll now hand over to Dr. Jason to begin the session.
Thank you so much, Ms. Leshna, for the warm introduction. Good morning, everyone. Alright, so it looks like I haven't got our coffee or breakfast yet. Fantastic. Hi, my name is Dr. Jason. I'm from the National Cancer Society, NCCM. Before we start this research track and hear from our wonderful speaker, I just want to share a quote from a previous NCI, National Cancer Institute, and a Nobel laureate winner. If you want to do something truly important, something that makes a difference, work in cancer research. This is by Harold Varmus, the Nobel laureate and former director of the National Cancer Institute. He discovered proto-oncogenes and oncogenes as well. So I think we're all in the right track. If you're on cancer research, we are truly doing something. So without further ado, I'll introduce our first speaker for today. Prof. Felicia Chung Fei Lei. Sorry, I butchered your name. So the epigenetic edge, the title of her topic will be the epigenetic edge, epigenetic biomarkers for cancer monitoring and risk stratification. Dr. Felicia Chung is a dedicated cancer cell biologist with a strong background in molecular biology and bioinformatics. Her research centers on cancer epigenetics and cancer cell biochemistry, supported by competitive research grants from agencies in Malaysia, Japan, and France. Dr. Chung has led and contributed numerous high-impact studies on drug repurposing, immune evasion, epigenetics, metagenomics, and cancer signaling with publications in leading journals such as Nature Communications, Nature Reviews Cancer, Environmental Health Perspective, and Nucleic Acid Research. She currently serves as a program leader for BSc Horn's Medical Biotechnical Technology program at Sunway University, where she is passionate about teaching and mentoring students. Beyond academia, Dr. Chung is actively involved in initiatives promoting early cancer screening and contributed to research and community activities aimed at raising awareness, funds for cancer research. Her work reflects a deep commitment to both scientific advancement and public health. Without further ado, Prof. Chung.
Hello. Okay, thank you very much, Dr. Jason, for the kind introduction. It is really my great pleasure to be presenting here today, sharing the stage with people that I idolize as a researcher myself. For today, I will be speaking about just a few little topics covering something close to my heart, which is cancer epigenetics. So I'll start off for the benefit of the students in the audience, maybe, or those who are not so familiar with epigenetics, with an overview of epigenetic biomarkers, before I speak a little bit about our work. Okay, so just briefly, what is epigenetics? It is the modification of DNA which leads to a change in the expression of genes that is both semi-permanent, so heritable, but also reversible. So this is the very important part about epigenetics, is that it's potentially reversible. So if we find an epigenetic mark in cancers that lead to a cancer phenotype, we can potentially reverse it back and restore the cell's original phenotype. Now, there are several modes of epigenetic modifications, but the most well-known and well-studied one would be the DNA modifications, that's your DNA methylation. Your histone modifications, those are your histone acetylation, histone methylation, also your non-coding RNA, which are your siRNA, microRNA, and more recently, RNA modifications. So those are like methylation on the RNA, which leads to changes in the stability of the mRNA. So how are epigenetic biomarkers useful in general? So epigenetic biomarkers, and I'm sure many people in the audience, or even in the booths outside, those like, for example, Gene Solutions, they are very well established as early cancer detection tools in recent years. So these are, for example, microRNA or cfDNA from patient serum has been used in early cancer detection tools, and also as biomarkers for predicting for recurrence and response to therapy. So I'm just going to run through a few landmark studies over the years showing how epigenetic biomarkers have evolved. I'm going to start out with a very early study from 1998. Here is an early one where the researchers of this PNAS paper showed that P16 methylation could be observed in hyperplasia, adenomas, and adenocarcinomas in tumors that were induced by NNK in a rat model. So for fun fact for the students in the audience, nowadays if you try to present a stitched gel such as this, you might get into a little bit of trouble with your supervisor. But back in the day, it was quite well accepted. So here we saw that there were increased methylation in the adenocarcinomas and very low levels in the hyperplasias, but these sites was not methylated in the normal cells. So critically, these researchers, they showed that the increase in methylation was seen in squamous cell carcinoma, and they could also be observed in the sputum of individuals who were not just with lung cancer, but also those who were cancer-free but were also smokers. So this started really a wave of studies in cancer epigenetics, and I cannot speak about cancer epigenetic biomarkers without mentioning SEP9. This is one of the most famous cancer biomarkers in epigenetics. SEP9 is septin, the gene septin9, and methylation of the septin9 promoter has been observed in over 90% of tumor tissue compared to normal colon tissue. And in a large study, a prospective study with almost 8,000 participants, they observed that the increased methylation of SEP9 could be observed in individuals with CRC. This is in the blood of the patients, and the sensitivity was not so great, 48.2, but specificity was 91.5. So the first ever blood-based screening test based on epigenetics was based on SEP9. So this would be the EpiPro-Colon test, which became which is currently available and was approved by the FDA in 2016. So all is great so far in epigenetic biomarkers, but then came the CCGA project, which basically blew everybody out of the water, and blew everyone's minds away, because they were the group that really did a comprehensive profiling of the cfDNA from thousands of individuals. So they started out with a whole genome bisulfite methylation analysis in the cfDNA, and they developed a test which had a 99% specificity and could accurately detect tissue of origin. So in this test, they could use the epigenetic patterns that they observed. Of course, they did not disclose which exact patterns those were, to accurately determine the site of the patient's cancer based on the blood test. So this was, of course, one of many publications that eventually led to the GRAIL test. It's famous for many different reasons. Okay? But it's not only in the Western population. We also saw a paper quite recently emerged from China. This was from the Pedro longitudinal study involving more than 100,000 healthy subjects, where the authors, based on, in fact, they started with a literature search and the CCGA data. And they started out by identifying genomic regions that were significantly differentially methylated between the cancer and the normal tissue. And from here, they developed what they call the PANSEA test based on 477 genomic regions. And they tested it out on their samples, which were collected from apparently healthy individuals. And they observed that their test was able to detect the 5 most common types of cancer with 88% specificity in post-diagnosis patients. So these are individuals which already have cancer, and with a specificity of 96%. And in those who were asymptomatic, so these were what they call the pre-diagnostic cancers over here, they could detect cancers in 95% of them. So they basically did a logistic regression using these genomic regions. They developed a logistic regression score, which they called the LR score on the y-axis over here. And they could see that in healthy patients, healthy individuals, the methylation LR score was low, and then the LR score was high in people from post-diagnosis and up to 4 years pre-diagnosis. So when they divided it based on cancer type, as shown in this diagram over here, they saw the same thing. They could see high LR scores in all the 5 common types of cancers, and this is the post-diagnosis, and this would be the pre-diagnosis samples. So it was quite compelling data out of the Pedro longitudinal study, showing that epigenetic biomarkers are indeed fairly promising early biomarkers for early cancer detection, and it was demonstrated in the Chinese population. Okay. One interesting thing that they saw was that the earliest case they found was where the LR score was high in an individual 4 years prior to diagnosis. So before I move on to our work, I just want to highlight one piece of work from a former group of mine. Here they were using epigenetic biomarkers to predict for recurrence or response to therapy. In this study, they took the tumors, they didn't take the tumor cells, in fact, they were working on oral cancers, and they were looking at the surgically negative regions of individuals who eventually developed local recurrence compared to those who didn't develop local recurrence. So an interesting thing about this study really is that they were not looking at the tumor cells themselves, but in fact the negative regions that were surrounding the cancer cells. So they found a panel of 14 CPGs that could predict for local recurrence in oral cancers. And what was interesting is that when they tried to replicate this finding using the TCGA data, they were able to replicate it. So this 14 CPG marker could predict for recurrence in the oral squamous carcinoma. So there are indeed many examples of similar studies, but I think this is one approach that we could potentially utilize in our research in local cancers or cancers that are more common in the Southeast Asian region. So just a little summary of what I just spoke of, which was mainly what the literature has shown so far. There is utility for using epigenetic biomarkers in multi-cancer early detection. And epigenetic biomarkers can be predictive for recurrence, and so much has already been done. You might be asking, is there still more that we can still do? Okay, so I'm going to speak about 2 studies that we recently published. The first would be this one that was published in Clinical Epigenetics, where we were trying to investigate if there is a potential for using DNA methylation patterns in peripheral blood to predict breast cancer risk. Okay. So here we were using prospectively collected blood samples, the median being collected about 8 years prior to diagnosis of breast cancer. And we had 225 tumor tissue and also 27 tumor adjacent tissues. So here we wanted to look for epigenetic biomarkers in the blood samples and to determine if they could also overlap with the differentially methylated sites when comparing the tumor tissues to the adjacent tumor adjacent tissue. Okay, so this is a rough flow chart of what the study looked like. It utilized samples from the Epigenetic cohort. So there were several thousand samples that were collected in the 90s, and then we had, how many pairs was it again? I put it 340 pairs of blood samples. So these were age matched and also matched for menopausal status. And here we wanted to identify if there were differentially methylated sites between the cases and controls in the blood samples and also in the tumor tissue. Okay, and fun fact, they didn't really overlap. When we tried to overlap the differential methylation between the tissue and the blood, the overlap was only about 2 sites, and the direction of the epigenetic change was not the same. So what we concluded from this, and I'm not really going to show the data here, is that actually the epigenetic changes in the blood and the tissue in this cohort particularly were not in overlap. So unlike some of the other studies that I highlighted before, where the methylated DNA was slowed off from the tumor tissue or were derived from the tumor tissue, we think that in our cohort we didn't see the same source of DNA. Okay, so using the blood samples, these 340 pairs, we did model training and validation to develop a classifier to see if we can predict for increased breast cancer risk. And what we saw was that there were some differentially methylated sites between the cases and controls in the blood samples. And for these 187 of them to be exact, for just about a third of them, 75 of them, we found that they were significantly correlated to the time to diagnosis for that particular patient. So for example, here I'm showing SIRF6, one of the sites. It was lowly methylated in those who were closer to the time of diagnosis, and for example, in the controls, this site was hypermethylated, all right, and then it became hypomethylated in those who eventually developed breast cancer. But for those who were closer to the time to diagnosis, this hypomethylation was more pronounced, and then we saw this also for REXO1 and a separate site that I'm showing over here. So how did the classifier go? So we developed this classifier based on PAM machine learning algorithm, and we found that the AUC for this particular predictor was 0.86 when we tested it out on a validation set. This set, of course, was never trained with the original model, and then we just to make sure we're not seeing any mathematical mumbo jumbo, we did label flipped controls, 500 of them, and we ran the same machine learning model to determine that the prediction was not due to any biases in the data set. So as expected for the controls, the average AUC was 0.51. So we were fairly confident that our model is indeed predicting for early breast cancer risk. So when we look closely at the predictions in this test set, there were 68 pairs of cases and controls. We saw that most of them, the model predicted correctly the observed case control status. But for those where it predicted wrongly, we were curious to see who these individuals are. And if you note from the time to diagnosis over here, more than half of these individuals that the model predicted wrongly were diagnosed in the 3rd to 4th quartile after the sample collection. So these were individuals who were diagnosed more than 8 years after the sample was collected. Because as I mentioned before, the median for time between the sample collection to diagnosis was 8 years for this particular cohort. Okay, so that's what I wanted to share from this study. The second study I wanted to share is something that we recently published in Nature.com. Here we were looking at small intestinal neuroendocrine tumors, I call them SINETs. These tumors are quite interesting in the sense that they show very low mutational burden. This is known in the literature, and they have a notable lack of somatic driver mutations. But one interesting thing that about these kinds of tumors is that they can be multifocal or unifocal. So multifocal being that they are multiple tumors in the same patient. Okay? So here we did multi-omic profiling of 219 samples from 122 synapse patients. The aim here is to try to identify molecular subgroups of synapse because historically these synapses have been treated as one entity. Okay? And we wanted to identify molecular predictors of multifocality. So what is it that differentiates the unifocal from the multifocal disease? I see I have 5 minutes, so sorry, I'm zooming already, but I'm going to zoom even more. Right, so the results from the genomics analysis was that indeed it has these kinds of tumors have a low mutational burden as we expected. We observed some chromosomal aberrations, okay, but they did not predict for multifocality or unifocality. Then we went to the transcriptomics. So based on the transcriptomics, we did the typical thing that many people do nowadays, doing molecular subtyping using the transcriptomic data. And from here, it was classified into 4 different molecular subtypes. Running out of time, I'm not going to go into too much detail, but the main point here was that there was one subtype, which was called the mesenchymal subtype, which demonstrated poorer survival compared to the rest of the subtypes. But again, with transcriptomics, it did not clearly separate the multifocal to the unifocal disease. And then we went to epigenomics, which is my baby. Then in the epigenomic data set, we again identified 4 subtypes. We tried to see if it overlapped with the transcriptomic subtypes, and we saw that it did to some extent. So there are, for example, there's one subtype over here, which we call the epithelial enriched, that was comprised mostly of the epithelial subtype from the transcriptomic analysis, but you will notice that it's not exactly matched, all right? But the critical thing, does it predict for multifocality? The answer is yes, it does. In fact, it was the only one of these analyses where we could see a difference or significant association between the subgroups that molecular subgroups and multifocality versus unifocality. So yeah, we called what we call the hypomethylated subtype had an enrichment of the multifocal disease, and the one that we call the unifocal enriched subtype, as you expect, has enrichment of unifocal disease. Okay, but this part of the results are not yet published. So here, using the epigenomic data, we wanted to see if there is a difference in the epigenome between the tumors from the same participant. So remember I said that some of these participants have multifocal tumors. We profiled several tumors from the same participant. And here you are seeing on the right-hand side the unifocal participants, and on the left-hand side you see a group of the multifocal ones. So here I'm showing the results from the top 5 most differentially methylated DMRs when comparing the multifocal versus the unifocal. And in the unifocal group, you will see that they are mostly hypermethylated, and here you will see that there is a difference between the unifocal and the multifocal. Yeah, that's the point of doing of what we ran over here. But when you try to look at the DNA methylation levels of the different tumors, even within the same participants, so this is one patient, you will see that there is some variation in the epigenome, even within the same patient. So we found this quite interesting, but it was very complicated to explain in the context of the whole paper. So currently this is something we are still exploring to see where it goes. Okay, so skipping through, so just a take-home message for my talk, basically, that epigenetic biomarkers, even though a lot has been done, there is still a lot to be done. In our work, we determined that epigenetic biomarkers can refine the classification of synapse, and it can also be a determinant of multifocality. And I sure hope that we can apply this approach to studying cancers that are more common here in Southeast Asia. So seeing as I only have 1 minute left, I'm just going to spend the next 1 minute to thank all my friends, collaborators, and students who have done all the work. And I want to take this opportunity, since this is a conference that brings together people from all spectrums of the cancer care continuum. I just want to say to especially the young people in this group that sometimes collaboration doesn't always have to be academic. Sometimes we can support each other even through non-scientific collaboration. So here are all my cute little students and also my collaborators over the years. But you will notice that in some cases, we support each other in ways that are not scientific. So here, we have Sunway Group sending 100 runners to last year's Relay for Life. Yeah, and we have also been supported. Here is at the Nasopharyngeal Carcinoma Society of Malaysia, supporting us in our own run, and Magna supporting us in our run, and us supporting Lung Cancer Network Malaysia in all their activities. So really, it is a collective effort together, and I hope that everyone takes away that we can work together. And sometimes it's just a matter of showing up for each other. Thank you very much.